The next lecture is going to be on quality control and normalization. So you noticed I've been talking about log normalized counts and scaled counts. So now we're going to talk about where that comes from. Why do we do log normalized? What does log normalized counts even mean? So this is the overall picture that you're going to learn by the end of this course. Possibly with the slight exception of trajectory inference. We're not going to talk too much about that unless someone really wants us to. So put it on Slack if you want to talk about trajectory inference. We're going to do clustering and differential expression. And we're also not going to, we'll talk about validation follow-up, but obviously we won't do validation and follow-up. So the first thing we're going to do is get our normalized expression. And then we're going to do all of these other steps till we get to clustering, and then that's going to be when we have our different cell types. So we're starting right at the start here today. So for quality control, our goal is to remove dead or damaged cells from our data set. So cells are sensitive. When we dissociate them to put them on our 10x machine, they can get damaged and die, or they might get so stressed they can start committing to apoptosis and kill themselves. Unfortunately, even though we do our empty drops filtering and our cell ranger filtering, some of these cells that are still damaged or super stressed and undergoing apoptosis will be included in our data set, even after that filtering. So we need to figure out how to get rid of these. So all we have, right, is our UMI count matrix. So we can look at how much is each gene expressed in each cell, and that's all we know. What statistics could we try to calculate from that to estimate the cell quality? So Ottawa suggests using total reads. Yeah, we could absolutely use the total reads or the total UMIs, right? The total amount of gene expression we detect. Yeah, Ottawa also suggests the expression level of apoptotic genes. Right? We know genes involved in apoptosis. We could look at the expression of those. Got another suggestion for mitochondrial RNA. So when cells get damaged, they release the RNA from their mitochondria. So we could also look at that. Yeah, the total expression per cell. So that's definitely some of them, but there's lots. So I've looked at, put these in some general categories. So we could look at apoptosis-related genes. We could look at housekeeping genes, right? Those are genes we should detect if it's a good cell. We could look at ribosomal genes, mitochondrial genes. We could look at metabolism genes to see if that cell is still actively doing metabolism. We could look at mapping quality, or we could look at things like whether we see marker genes of different cell types expressed. How many genes are lowly expressed? How many total reads do we see? How many different genes do we see? Etc. So there's lots of different potential things we could calculate. So how do we know which statistics we should use? So for this, we're going to go back in time, all the way back to 2014 or 2018, when we were using the Fluidigm C1. So this is a microfluidic device. And the reason we have to go back to this point is because this microfluidic device allowed you to actually take pictures of your cells that you had captured. So you could actually see if there's an intact cell there or not. So you could see if it looks damaged or if there's nothing there to check it for your RNA-seq data. was there actually a cell there? Was that cell damaged? And then you could test what statistics can discriminate between those cells that are dying based on the image in my RNA-seq data. So they took all of these different scores and compared them for their ability to predict whether the cell was broken, whether there was multiple cells captured at once, or whether there was nothing captured at all. They tested all of them and came up with 2 that work well. So this is the mitochondrial RNA proportion, which was the best for detecting broken or damaged cells, whereas the total number of detected genes per cell was the best predictor of whether there was just no cell present at all. That is why we still use those two statistics today for calculating these things. Lots of people make up reasons, rationales for why we use them, but we actually use them because we tested all the possibilities and these 2 work the best. So when we're filtering our data set, we're going to look at the distribution of these 2 measures across our cells and filter out those with poor scores. Okay, so that's quality control. The next is normalization. So to understand why we need to do normalization, you have to understand the problem of library size. So we're doing our sequencing. We assume the chance of sequencing any particular molecule in our sequencing library is equal. But what would happen if, for whatever random chance, we happen to double or quadruple the amount of sequencing for a particular cell? So if we double the amount of sequencing, yeah. So if we double the amount of sequencing, you auto got it, that we would have twice as many UMIs detected, more or less. So if we go to compare our single-cell RNA-seq data, right, this whole distribution up here, not even looking at this lower secondary knee, right, this line here covers 2-fold of total UMI counts per cell. Right? At this end, we've got more than 200,000 UMIs per cell, and down here we've got only 100,000 UMIs per cell. So if we were to try and compare those cells to each other, right, a cell from over here, all of the genes are going to be twice as highly expressed. We're going to have twice as many UMIs per gene than the red cell. So if we tried to compare them, we would say blue cell has upregulated every gene in its genome 2-fold. But that's clearly nonsense. All right, that's just because we happen to sequence more UMIs for blue cell than for red cell. So we want to get rid of this effect of total UMIs per cell. So the simplest solution is what's known as counts per million. It's called per million because when you're doing this for bulk RNA-seq, you do actually do it per million reads. For single-cell RNA-seq, we don't do it per million, but we still call it per million for historical reasons. So here we simply take the expression in each cell, we calculate the total UMIs in that cell, and we divide the expression by that total. So we essentially get the percent of all of the RNA in that cell that came from each of the genes. And then we just multiply by a big number to turn those percentages back into normal-looking numbers. And so if we do, because we have 10,000 genes across the genome, if we do the percent of expression that comes from each of those genes, it's going to be very, very small, okay, like 0.001%. We could computationally deal with that just fine, but it's annoying to look at and to tell someone, right? You don't want to say, oh, the T cell receptor was expressed at a percentage of 0.0005. It's just annoying to tell people that. So we just multiply it by 10,000, and that turns that 0.005 into 50. And say, okay, we see the expression of the T cell receptor at a rate of 50 counts per million. So that works reasonably well, but we have this problem that of proportionality. So if this was our single cell, and if we pretend we only have a small number of genes, and in one condition, so let's say this is our normal case, we have 5 reads for gene A, or 5 molecules of gene A. If we then go to a different condition in disease, let's say, maybe in cancer, now we have 50 copies of gene A. If we then calculate the percent of total RNA for each gene and compare it between each other, you'll see that all of the other genes look like they've gone down in expression, right? Their percentage has gone down from the total RNA, even though the actual expression level hasn't changed, right? It's just that one of the genes has changed a lot. So we've had a few different proposals for how to fix this problem. So most of the time we assume that most genes are not strongly differentially expressed between our cells. And then we can use approaches, we could try to use approaches that have been developed for bulk RNA-seq, like D-seq, D-seq size factors, or EdgeR's trimmed M values. I'm not going to really go into that because these don't actually work for single-cell RNA-seq because they assume every gene is detected in every cell, and that's definitely not the case in single cell. However, we can use them by using a trick in a method called scran, which pools together cells so that we do have expression of every gene in every pool, and then we can calculate our D-seq2 size factor or our trimmed M value on that pool. And if we do that multiple times over and over again, we can mathematically calculate what the size factor should be for each cell. That was the state of the art for a while, until SC-transformed was created by the creators of Surat. So they noticed that not all genes behave the same. So here, each of these plots is looking at the expression of a gene on the y-axis, or a group of genes on the y-axis, versus the total expression in the cell on the x-axis. So you see we have a positive relationship where cells with more total UMIs have higher expression of all of their genes. And these are pooled from most highly expressed genes up here to most lowly expressed genes down here. So this is what happens if we use the counts per million normalization and then log transform those values. So you can see for these genes, we have successfully gotten rid of that relationship between total UMIs per cell and the expression of that gene. Same for these guys. But for these more highly expressed genes, we haven't fixed it. Right? And same for the most highly expressed genes, we haven't fixed it. We still have cells with the highest total UMIs have the highest expression of that gene. Right. So in order to fix that, they proposed using the negative binomial distribution because single-cell RNA-seq data, the raw counts, follows a negative binomial distribution. This is the properties of that distribution. I'm not going to go through it too much in detail because it's very mathematical. There's a nice paper in Nature Biotechnology down here that proves that this is the distribution that single-cell RNA-seq follows. So if you want to look at proof of why this is, you can look at that. But otherwise, I'm just going to say we're going to accept that this is the case. So what SC-transform does is it takes that negative binomial distribution and it takes all of the genes and bins them by the expression level and randomly chooses a few genes to represent each bin. And then it performs a regression on the total sequencing depth versus the observed expression of that gene, of those genes, using the negative binomial distribution. That estimates what that relationship is, then it uses those few genes where it fit those values, and it extrapolates them to the rest of the genes across the whole genome. And then it just calculates the residuals from that relationship. Right, so we're taking a linear regression and we're calculating the residuals from that linear regression. And that becomes our new normalized expression values. So it specifically calculates the Pearson residuals, just calculated like this. So we have our observed counts minus the expected counts given the sequencing depth of that cell divided by the square root of the variance. And if we do that, then you can see here's our slopes before, as I showed before, where with log normalize, they're not centered at 0. We still have positive slopes for more highly expressed genes. If we use this Pearson residual or SC transform approach, all of them pretty much are now around 0. So it corrects for all of the different relationships for the genes at different expression levels. Which is why this is now the go-to normalization method in single-cell RNA-seq. The other advantage of it is because it's using a linear regression model, you could correct for other things as well. So if you want to correct for the quality of the cells, you could add something like the proportion of mitochondrial expression or the total genes detected per cell. You could also correct things for like batch effects. So if you have samples from multiple batches, you could add that batch label to the regression model and it will correct for that at the same time. Okay, so going back to the norm of normalization. So the goal of normalization is to make our data follow a normal distribution. Because many statistics assume our data follows a normal distribution, not a negative binomial distribution. So in general, our options are either to redo all of the math for our statistics, now using a negative binomial distribution, which is hard, or make our data look like a normal distribution. So here's our raw UMI counts. Here's our counts per million normalized data. If we log transform that, you can see we now have kind of a normal distribution with this one spike down at 0. And if we use the Pearson residuals, we also get a more normal distribution-like distribution. So because of that, this will allow us to use methods like principal component analysis that assume our data follow normal distribution on our single-cell expression data. We can also look at different confounders. So these are biological differences between our observations that is not our variable of interest. So this could be things like the cell cycle, the genetic background of different patients, right? If we're doing patient case versus control, how we stored our tissue, cell stress, the age of our different patients, or how long our samples have been in the fridge, right? That can affect our gene expression, but it's not something we care about. There's a bunch of different potential solutions to these. So the best one is always to design your experiment so you don't have that confounder. So if you can do that, you should do that, but it's not always possible, especially if you're working with clinical data from patients, right? There's lots of constraints on how you collect that data that you can't get around. You could use regression models to remove it. So something like how long that your sample has been in the fridge, you could add that to your SC transform regression model to correct for that. In many cases, you can simply exclude the cells or genes that are most affected by it. So that's typically done for things like cell stress or the cell cycle. And so if you have cell cycle affecting your data set, maybe you just want to exclude all the cell cycle genes. Or if you have a lot of stress in some of your samples for particular cells, maybe you just want to throw out those cells that are really stressed because they're not giving you useful information. You can also include this as a covariate in regression models that I'll talk more about in our differential expression section. Or what is also still very popular is just ignore it and hope it doesn't matter. And then you just do lots of replicates and hopefully you get the same answer across all your replicates, and all those confounders are not driving any of your problems, any of your results. So for the cell cycle, there's lots of options for dealing with the cell cycle, but you have to be a little bit careful because things like regressing out the cell cycle will also remove any variability that is associated with your cell cycle.
Before we move on too far, I just want to make sure this question gets answered. So by using smaller subsets, does the central limit theorem apply?
Does the central limit theorem apply? The central limit theorem does not apply because the central limit theorem is talking about averages most of the time. It can also be talking about sums, but in general for our single cell RNA-seq data, the central limit theorem doesn't apply because our data is it follows a negative binomial distribution. It doesn't follow a normal distribution. And we're generally not doing averages when we're looking at raw expression values. If you're looking at something like the average expression of a gene in a cell type, then if you have enough cells of that cell type, then the central limit theorem does apply.
Okay, great, thank you. And I see that there are a couple other questions coming in that it might be worth addressing before we move on.
Yep, I'll pause. So we have another question from you, Ottawa. Can we consider covariates for normalization in SC transform? For instance, studying the design matrix. Yes, so you can include lots of covariates for normalization in SC transform. In theory, it works well. In practice, we've actually noticed the regression function of SC transform for things other than library size don't necessarily work all that well. We're not entirely sure why. We've just started testing it and we've seen that it does correct for it, at least in part. But there are other ways of correcting for covariates that might be better than SC transform. It probably has to do with the fact that they're extrapolating from a relatively small number of genes and a relatively small number of cells. So SC transform also subsets cells so that it runs faster. So we think that might mean it's not actually that great at calculating the values for some of the covariates. Another question, what analysis can be applied if I'm interested in knowing the effects of cell cycle and apoptosis? So if you're interested in knowing the effects, then you don't want to regress them out. You want to use them in your differential expression test, which I'll talk about tomorrow. Okay. Looks like all the questions online from Ottawa. So for the cell cycle, you want to be careful because sometimes the cell cycle is actually of interest. So particularly if you're looking at something like cancer, which cells are and aren't cycling might be an important biological finding. So in that case, you don't want to regress it out and erase it from your data set. If you're looking at mature tissues, hopefully most of your cells are not cycling. So you usually don't need to worry about the cell cycle in mature tissues. If you're looking at development or differentiation, that's a case where the cell cycle might be a confounder that you might want to regress it out. But again, it depends whether you're interested in which cells are most actively cycling and which ones aren't, or whether they're just all cycling and you just want to get rid of that confounder so you can see the developmental trajectory. Okay. So moving on to the next quick topic that I'm not going to delve too much into, but there's lots of methods out there that impute or smooth the data. claiming that this makes your data much better and your findings much better. I'm going to respectively disagree with those and say you should be very careful about imputing or smoothing your data. Because here's just a little toy example where half of the genes are differentially expressed between two conditions. So some of them are upregulated in red, some of them are downregulated in blue. And this box here is the gene-gene correlation matrix. So you can see all the upregulated genes are correlated to each other and anti-correlated with the genes that are downregulated. And then I have a bunch of genes here in gray that are not different between my conditions. They're uniform across both conditions. Then I ran a bunch of imputation methods on it. And you can see now suddenly a lot of my genes that are not different between my conditions now are different. Right, so all of these guys in gray, and for magic, they're now all completely different between my two conditions. Same with using this DCA method. And this KNN, it's not as bad, but you can still see lots of these genes that are not different between my conditions are now magically different between my conditions. So these are false results and will give you false conclusions. So in general, you should never run any sort of statistical test on imputed data because it will give you false results. So if you want to use imputation, it should only be used for visualization. And just be aware, if someone else in a paper is using imputation, their visualizations will look way better than they really are because they're using this smoothing to make their data look artificially good, essentially. Okay. So any questions about these sections? I don't see any.
Imputation is only used for publication purposes for image?
Yeah. Yeah, so you can, people do use it for visualization in their publications to make their results look amazing. I'm personally still not a fan of doing that, but if you want to use imputation, you can use it for visualization without causing your results to be false, right? You're only using it to visualize after you've done your statistics to show the patterns you see, and it does make the patterns much more obvious. But you should never draw conclusions based on imputed data, because it's very likely those will be false. But if you want to do it to show off, look how amazing the trend is we saw, you can sometimes fool people. All right, so to summarize, for QC, you're looking at low genes detected per cell, indicates a dead cell or an absence of a cell. High percentage of mitochondrial indicates a damaged or stressed cell. Normalization is often necessary for downstream analyses, especially things like PCA. Bulk RNA-seq normalizations don't work for single cell, but we can use SCRAN as a trick to get around that. There's lots of potential biological confounders, and there's no gold rule for how you deal with them. It depends on your particular system and question you have, and how good your data is and how impactful those confounders are. And if you're going to do imputation, use it for visualization only. So that's the end of this section.
30 minutes next week.
Okay. So we will plow forwards and go to our lab practical.